Hi there, I'm Patrick Degener and this is bioelectronics. In this lecture, we're going to talk about interneuron transmission, how are signals transferred from one neuron to the next. So the key topics for discussion are types of the need for transmission, types of synapses, the connections between neurons, chemical, electrical, and also their geographic locations. And then we're going to talk in detail about how that synapse, the chemical synapse operates, and then we're going to talk about different types of neurotransmitters. So let's take a conceptual process. This is when I was young and didn't have quite the same belly I have nowadays. And what this scene is describing clearly is an action scene. And what I like about it is that you can see that I'm sensing a certain aspect, where the individual's head is. I'm sensing in terms of the kick that's landed on the head. I can sense the touch. Now the action, so this all goes to a processing center, which is my brain, and then there's actions arising from it. Now clearly there's physical actions in terms of the movement of a leg in this kickboxing arrangement, but also in a whole number of other forms, such as for example, sweat reflexes to counteract overheating. So these actions, the first action is a conscious action, moving of the muscles, and then there's an autonomic action, which is the sweat reflex, and of course there's things like heartbeats and fear and all sorts of other responses going on. So there's many other examples of this: people playing tennis, driving cars, walking, hunting, and a whole number of other senses. So the key aspect here is that there has to be communication from this center, this processing center, which we call the brain, all the way down to the extremities where we have touch sensors or perhaps muscles that we need to activate. And this distance can be a meter, 1.5 meters. And if you think about giraffes, this distance can be 5 meters. This can be quite an extreme distance. And if you go as far as the dinosaurs, sauropods, perhaps that distance was as much as 14 meters, perhaps even longer. So clearly there has to be a connection from the processing center to these extremities, and signals need to be passed between them. And these are sensory signals to the brain and action signals from the brain to those extremities. So in terms of transmitting information, we've described before that we have, the primary form of information is something called the action potential. And the reason for the action potential the way it is that because it has a digital amplitude, as it traverses over distance, it does not lose information in terms of height. So information is passed by the action potential, which jumps, in these motor neurons, jumps past these myelinated cells until it gets to the end. But then, of course, once it gets to that end point, it has to connect with the target. Now that target may be the sweat gland that you saw in the previous picture. It may be the muscle, e.g. the leg muscle, or it could be another neuron. And one thing to point out is that when we're walking, we don't consciously go left foot, right foot, left foot, right foot. That would be problematic in a sense that you wouldn't be able to think other things and talk to people and multitask that, at least some people are very good at doing. So one of the components within our spinal column is something called a pattern generator, which contains some basic memory of left foot, right foot, left foot, right foot. So a signal can go down from the brain saying walk, and your reflex, which emanates from these central pattern generators, your reflex can sort of keep you going until you need corrective steps, or you trip, or you need to adjust your particular movements. Another point to make is that transmission is not just between the brain and the extremities. Transmission can be also local within the brain. You could have one brain region that needs to communicate with another brain region. So, in order for neurons to pass information to each other, there are something called synapses. So we've discussed before about the operation of a neuron in terms of creating the action potential. Signals traverse in, down the dendrite, and signals are then integrated, and if they pass a threshold, then you get action potentials down the axon. So this is the input, the dendrite, and the axon is the output of this particular cell. So then, of course, there have to be connections. Now, the connections into the cell and connections from the cell to other cells, or maybe glands, muscles, or neurons, this is called the synapse. And the objective of the synapse is really twofold. Primarily, it is to transfer information from one place to the other, but it also has a function in computation and memory, as we'll see a little bit further on. Now, there are different types of synapses. You've got synapses which can connect to another cell's axon. You can go from an axon in one cell to a dendrite in another cell, so axodendritic. You can connect directly to the soma, so this is axosomatic, and of course you can actually connect to another cell's synapse. So if there's another cell here, you can connect to the synapse and perhaps prevent or enhance that synaptic connection. And unlike transistors, where effectively you've got three connections in these devices, neurons can have multiple connections in and multiple connections out. And a way to perhaps simplify this approach and to simplify neuron circuits is to think of neuron circuits in this very, very simple model. So you have inhibitory cells and you have excitatory cells. E for excitatory, I for inhibitory. And this is a little bit like sort of transistor circuits having P-type transistors and N-type transistors. Now, the inhibitory cell can have inhibitory connections to the excitatory cell, or it can have inhibitory connections to itself. The excitatory cell can have excitatory connections to itself and excitatory connections to the inhibitory cell. And of course, this is a very, very simplified route. There are lots and lots of different cell types. But in essence, neuron circuits consist of synapses that try to pass positive information, excitatory synapses, and then you've got connections between cells, synapses, which try to inhibit the activity of the target cell. So this is like a push and like a pull. And you can argue that some of these computations can add to, at least conceptually, a little bit like the the 1 and the 0 in digital logic. But again, we'll talk more about this in future slides and lectures. So in terms of connections between different types of neurons or neurons and other cells, there's really two particular types. The first type is what is called the electrical synapse, and the second type is what is called a chemical synapse. Now, an electrical synapse is really just a very fast connection from one cell to another cell. The objective is simply to be the simplest form of wired connection. It's not trying to do any significant processing through that synapse itself. It's just trying to pass things through. A good example of an electrical synapse is the horizontal cells in our eye. And what horizontal cells do is they share information from the pixels, the rods and basically the cones. And what they do is to perform a function which is a bit like a Gaussian smoothing. So they kind of smooth out the noise. So by sharing information with each other, instead of having one pixel being noisier than the other pixel, it kind of smooths it, smears it all out. Reduces resolution, but it allows for a reduced noise. The more common type of synapse inside our body is the chemical synapse. Now, chemical synapses are slower, but they allow for a certain amount of computation. And if you think, again, I'll discuss this more in detail later on, but the key criteria for any synapse is how efficiently it transfers information from the pre to the post synapse. If it transfers it efficiently, we call it potentiated. If it's inefficient, we say it's depressed. And these connections, the synapses, can be tuned, with activity, again, that I'll describe in the next couple of slides. But that concept of potentiated and depressed synapses are the basis for computation in neurons. It's a bit like having if a potentiated is a bit like a 1, a depressed is a bit like a 0, except the key difference with digital logic is that there's no real 1 and 0. There's lots of little numbers in between. There's different levels of being potentiated, different levels of being depressed. Now, if we think about exactly how this works, well, as we've described before, if you have a cell, and that cell eventually has an axon that comes to this synapse, the cell takes in information and creates an action potential. That action potential travels down until it reaches the end of the axon and reaches this synaptic terminal. Now, at that point, this action potential causes the release of what are called vesicles. So these are little bubbles, if you like, inside the cell, and these bubbles migrate to the surface and they release a chemical called a neurotransmitter. So a neurotransmitter is basically a neuroactive chemical that traverses from the pre-synapse to the post-synapse. The neurotransmitter gets released, and then two things happen. The first thing that happens is that, there are enzymes inside the synaptic cleft that are constantly trying to get rid of it again. So this means that when you release a chemical, you get an initial spike of this release, but eventually it goes away again. The second thing that happens is that when these chemicals are available, these neurotransmitters, they connect up with chemically gated ion channels called receptors on the post-synapse. So once they connect to those receptors, the ion channel opens up, and then of course you get sodium and so forth sodium ions rushing into the cell, which then can get integrated in the soma of the cell to allow for action potentials to form. Now, there's not just sodium, chemically gated sodium channels. There's also chloride channels and others, which allow then, of course, for the negative effect, which is inhibition. And this is conceptually, a diagram of how this process happens. So if you imagine in time, you've got that synaptic cleft, and if you actually looked at the potential on the other side, let's say you've got an electrode connected into this very closely, just into that synapse. So this diagram here represents the synapse. What would happen in effect is that for a short period of time, as the neurotransmitters get released, the neurotransmitters will activate the receptors and the polarization will start to reduce. Remember, these are negative numbers, So instead of having a negative potential, you'll start getting a less negative potential. Now, that travels down to the soma, and if it reaches the threshold, then of course you will get then an action potential forming. But let's say for now that you aren't quite there. So once the action potential on the presynapse stops, after a certain period of time, the neurotransmitters are no longer released. The enzymes in the synaptic cleft will eat up the neurotransmitters, and the cell will start to repolarize again back to its original form. The same can happen with a inhibitory synapse, where now it's no longer sodium rushing in, but it could be, for example, chloride channels coming in. Because chlorides are negative, you get what's called hyperpolarization, where the target cell or the target neuron becomes even more negative, and that makes it harder for other synapses to activate it. And remember, there can be many, many different synapses to any given cell. It could be like 1,000 in some cases. And at this point, it's worth highlighting different types of neurotransmitters. So glutamate is a very, very important neurotransmitter in the brain. A lot of our processing's performed through the glutamate channels. For example, in our eye, most of the communication is glutamate based. GABA is kind of the opposite. So this is your classic inhibitory neurotransmitter. So this is kind of your opposition to glutamate. If glutamate excites, GABA will inhibit or prevent activity. So all of the actions in the brain can be push and can be pull. Acetylcholine is a really, really important chemical for our musculature. So basically our motor neurons, the cells or the nerve cells which connect from the brain towards our muscles, their main chemical is called acetylcholine. And then we've got other neuroactive neurotransmitters, dopamine, glycine, serotonin. So serotonin and dopamine are very important in our mood and our emotions. So for example, serotonin is a key chemical targeted in the antidepressant drugs. So that you have these serotonin reuptake inhibitors which prevent those little enzymes sort of destroying the serotonin that gets released, and therefore there's more serotonin, and therefore there's there's increased happiness, if you like, in the case of people with severe depression. And it's worth noting, and this is the final slide in this particular lecture, it's worth noting that synapses are not distinct or unique. It's possible for neurotransmitters to be released from one synapse and travel around and actually have actions on other synapses. And this is really, really important, and it's something that sometimes gets forgotten about, if you like, in some of the attempts to recreate the brain in silicon. So that's it for this particular lecture. In the next lecture, we will go through the process in more detail and start exploring computation by neurons. Thank you very much, and see you at the next lecture.